So, good morning, everybody, and welcome to Quantitative Big Imaging, first lecture of the spring semester 26. And yeah, well, I'll see if this thing works. Yeah, there it is. Now it should work. Yes. So, what I'm going to talk about today is generally about the course, some, say, administrative stuff, and then a little bit motivation about how we can use quantitative methods in imaging. And then I'm switching over to more technical side, what are images, where they come from, and also science and reproducibility, how it's connected with the imaging and image processing workflow, and finally also a little bit a hint on what's coming actually in the last lecture on workflows. The course webpage is on this link, and I sent you a mail yesterday where you got the link. So everything is on GitHub and nothing on Moodle, so it's in completely open material. And the lecture recordings, they will be on YouTube, and there will also be links if you follow this link here. There is a weekly plan, and there you have all the lecture notes, and also the recordings are uploaded or linked on that page. So this whole course is done using Jupyter notebooks. So these notebooks, they usually have some code that I'm running to produce the figures. And for that, I need to run this import part. So it's always in the beginning of each set of lecture notes. Lecture notes, they are both as Jupyter notebooks. If you prefer to use that one, then you can even test and rerun the examples I'm doing. Or else, I also produce a PDF lecture note, which you can also read everything. And in the notebooks, I have also embedded notes, and these notes are also then in the PDF lecture notes. So the notes you don't see during the lecture, but there is some added text to the different sections. So a little bit about the course. Who are we? Who are teaching here? Who are you? A little bit of what you expect or what you can expect from the course. Why the course exists, and then a little bit about. Oh, that was a little bit strange. Yeah, and then we have a little bit of course outline too. And first of all, who am I? I'm, well, as I said, maybe said or not said, I'm Anders Kestner. I'm a beamline scientist at Paul Scherrer Institute, working with neutron imaging. So not the most well-known type of imaging, but pretty nice anyway. And then I'm also lecturing here in this class. I have been algorithm developer at Varian Medical Systems, mainly towards the computer tomography reconstruction. Then before that, I was a postdoc here at the Institute for Terrestrial Ecology. Nowadays, it's what is it called now? Environmental Sciences Department. And before that, I got my PhD at Chalmers University of Technology in Sweden. And the topic was signal processing. So I've done a little bit zigzagging from being into more computer engineering, moving over to the signals, then now I'm actually working with physics. So I've done a little bit of everything. My email address is here, but I also have an ETH address, the one that I used to send the mail yesterday. And this is actually something I didn't update in the slides. I prefer if you use my ETH address, because then I have separated my mailboxes with the topics and I don't lose your mails. Then we have Daniel sitting back there. He will be the TA and helping you together with me doing your exercises, because there are some exercises which I will come to later. And he is right now a PhD student in the last year or closing. Yeah, in X-ray microscopy group at ETH and also at the Swiss Light Source at Paul Scherrer Institute. So, how are you? This is just an assumption, but they're a very varied background. We have had art history researchers and we have had computer scientists. So, and in between there are physicists, mechanical engineers, essentially everything what you can dig out around ETH and also other universities actually. Skills and complexities can be also very varied. Some have very little experience with images, barely seen any. Oh, well, actually, you made a lot of pictures, but anyway, doing any analysis on the images. Then the background, how you approach your data. There are different ways. One is very interactively clicking, marking out regions, and let the software take care of the task for you. Or else, some may even be pretty skilled already in scripting or writing programs for doing the analysis. Then, depending on where you are in your studies, the data complexity that you're working with can be very simple, could just be counting dots, or something pretty complicated like analyzing microscopic images or looking at the human body and other things. It's actually pretty wide applications of imaging, and the complexity varies also in how much data you have. Some only have a few images. That's pretty easy to work with. But if you start working with, say, terabytes of data, time series that you want to see the time dependence in different things, then you are in a different league of complexity. So, how will it work with all these variations? First of all, we have some assignments. The exercises, they are there to help you to understand what is done during the lectures, or actually get a little bit hands-on. And the idea is to understand the workflow. There will be some amount of Python. Most in the exercises is already prepared, so you get more or less the setup, and then you have to change a little bit. Saying that, Python is good to know, but you can live without it, even though if you want to work with the material fully, it's good to have some basic Python skill, or at least scripting skill in some sense. Some of the exercises you can actually do also with tools like ImageJ. It's not as fun. It's a lot of clicking and dragging, etc. You can do it, and you can see the same things. So if you don't want to work with the scripts, there is, for many exercises, also the way to work with ImageJ, for example. For those who don't know ImageJ, it's a very widespread tool for image analysis. So it's like Photoshop for science. And you can mark up regions, you can get statistics, you can get profile plots, you can get histograms. So you can get quite a lot. You can also do some scripting in ImageJ. And honestly, not my favorite, but you can do it. So, and yeah, then I said quite a lot about the exercises already, actually. So there is no requirement that you do these exercises, but I recommend to do it, because then you get a little bit more hands-on what's actually meant in the lectures. The notebooks, they are, as I said before, prepared for the exercises, and in the lecture notes, no, in the lecture plan, you have on the right side also the exercise to click on, and then you land on the notebooks. If you want to do advanced, you can of course also write Python, Java, or C++ code, but that will take quite a lot more time if you want to do things from scratch. If you would like to do it, you're welcome to. It's not really the recommendation, because you will spend more time than needed for it. Then we have a project. This is also optional, but again, recommended because then you can really get an idea on a real problem. And the real problem is something that you can ask me to give you, or you can take a part of your master project, PhD project, and we extract a sub-project that could be relevant in the size and time. And then you actually would get, say, free assistance on your project. So that is another way. And the other thing and reason to do it is that if you do the project, I can use your project as a starting point for the exam, which means you should know it pretty well already. So then you have a running start in the exam. Yeah. And then in the end, of course, there is also a presentation, which is actually also quite fun to see what other people have done. So a little bit more hands-on, seeing what can be done with image data. And that is in the end of the semester, I think 28th of May. And this is the course overview. Today, I'm talking about the introduction. Next week, it's building data sets. Then we come to image enhancement, basic segmentation, and following with advanced segmentation. Then from there on, we go into the analysis. So we're actually starting to measure things. And then going on to special cases of segmentation. Then there is actually Easter break, so then go search for eggs. And after that, we go on with statistics, how to do a statistical analysis and what to think, things to think about in that sense. Also looking into a little bit about uncertainties. And then comes two more applied fields, which is dynamic experiments. So we start having sequences of images, and then end of April, there is imaging with multiple modalities. So sometimes it's not sufficient to work with only one modality, say X-rays, but maybe you want to have information from a thermal camera at the same time, and how you can merge those two. And the last lecture is about actually computer engineering, how to work with large amounts of data. And then there are a couple of weeks between. I think some of the Thursdays are actually also holidays. And that is the time when you can work on your project. And during this time, you can make appointments with me or Daniel. And then we can have small sessions and discuss your problems or ways to solve your problem. I also have a long list of reading material. I don't say you have to read it, but there is actually some interesting stuff. Some are books, and that's way more than you can read during this time. But anyway, it's good hints on literature if you want to go deeper into some material. And a couple of books that I actually think would make sense to at least download is the John Ross Image Processing Handbook. Also, there's hands-on image processing with Python. They are quite good starting points for the work. The Image Processing Handbook gives a great overview of the different analysis types you can do with image processing. And then there is also there's morphometry with R. Can be relevant also in the shapes part of the lectures. All these books can be downloaded on the ETH library webpage, so it's worth downloading them. So let's go to the real topic. Our motivation for coming here is you have data, but usually in the workflow it's you get some beam time or some experiment time on some instrument, Very happy. You do your experiment, and then you got data. What to do with it? And very many what I see also among my users at the Beamline, they are not really trained into working with images. And the question is now, how can you extract some scientific information out of this data? And the data can be gigabytes, it can be terabytes even. And for that, you need a very good strategy. And that is why we are here now. And also, the whole workflow is you acquire the data, and this plot shows, in principle, level of abstraction and what steps you have to do to reach that. And the plot shows more or less that in the beginning, you only have data. You have images, which you can look at. They may be nice, they may not nice, which is actually often the case. So what you have to do is you get your data, maybe you do some kind of reduction of the data, then you may do some transformation, reconstruction, then comes some filtering, but still you only have data with more or less good quality or closer to what you want, but it's still only data. You don't know anything about it. And then you go into segmentation, then you have started dividing the image into different regions, and then you start taking off, and when you start doing labeling, then you start getting actually some knowledge about the data. And then you can do the measurements, and all of a sudden you have reduced the data from being a lot of images into only a few tables. So the amount of data you have has reduced radically, and you can start learning something from it. And then from the measurements, you can do classification and do some statistical inference, and then hopefully you will learn something from your data. So, how to proceed? You start setting up your wild image processing workflow. And well, in this plot, you do get your images, and then you do some fancy stuff, some magic stuff, and then you do some really complicated stuff, and well, some mathy stuff too. That's always good. And then afterwards, you recognize that ice cream is good for your bones. But well, that's just a fun one. But anyway, it can be a quite complicated workflow where you really want to have a very structured way of working with the data, and ideally, all that in particular the magic stuff here, that you have to tweak magic parameters between each image, that is something we want to avoid. In particular, if we want to go from one image where it may work fantastic, maybe on the second one too, but if you start working with tens, hundreds, thousands of images, you have to be sure that you don't have to tune the parameters every time, because then you will sit there till the end of days. And that is what we want to avoid. So we want to automatize the whole workflow of processing the data. The computer can work. That one can stand and humming for itself for a couple of days. I don't care, as long as I don't have to do it. So, the experiment life cycle. The first one, as I said before, you got some experiment time. You have to have a good plan how you perform your experiment to have it in the realistic range. Sometimes you don't know exactly the range. That's a little bit of testing, but anyway, you need to plan your experiment pretty well. Then the actual measurement can produce loads of data, and that loads of data needs to be managed. And with a few images, it's easy. But if you get into terabytes for an experiment, then you have to have a plan how you actually slush the data from one point to the other. Just bringing the data, several terabytes of data from the experiment site to your lab. Do you have a computer that can work with it? And so on. So that is a lot of work that you have to plan if you have large data sets. And then, of course, you come to the processing of the data. And again, you need a good workflow and plan how to actually come through all this data. Data science is A field that has helped a lot actually to make more structure in large amounts of data. Also the infrastructure that it provides is helping a lot. But still, you have to have a plan. And here is a little bar chart of how much time is spent on different phases of the work. And this is actually not the youngest one, and it's also, say, a qualitative plot, but estimated in 2000, in 2000, you spent some, say, 8-10% on experiments itself. Then you did the measurement, which took a long time because the detectors were slower, you didn't have the intensity that you have today, and so on. So the experiment was very time-consuming. Then you did some data management, copying the data and organizing it. And finally, you put some time on the processing. Interesting to see in this is that, first of all, the measurement time has reduced to almost a minimum, whereas the management of the data has grown tremendously over this plot. Also, the processing actually does a dip and gets less percentage. So, in principle, where you put the most of the time of analyzing your experiment data is managing data and analyzing it. And detectors, they get bigger. Some years ago, we had 1K, 1K detectors. Nowadays, we do 8K by 8K or even larger. And at frame rate that we could only dream about 20 years ago. So it's very fast to produce a lot of data. Here, for example, we have roughly 2K by 2K, doing 1500 frames per second. That gives us 8 gigabytes per second, and that is the rate of data that we produce. And typically, the analysis work toolboxes that we are working with, like MATLAB, Aviso, which is an interactive data analysis tool, Python, mostly they saturate already after 60 seconds at that data rate because you don't have the RAM in the computer even to handle it. So that is a problem that we can't keep up in the same pace, so you can't do real-time analysis of this frame rate. Different sources of images. We have X-rays, which then produces X-ray tomography, 8 gigabytes per second. Diffraction patterns, 30 gigabytes per second. And nanoscopium beamline produces 10 terabytes per day, and the files can be 10 to 500 gigabytes each. Can you imagine opening a file or accessing a file of 500 gigabytes? You need a fast hard drive, and you need to swap a lot. In optical imaging, you have the microscopy. It's quite modest compared to the X-ray imaging, only 500 megabytes per second. And we can also do some high-speed confocal images, 78 megabytes per second. Then we have all the consumer stuff. GoPro, this is not the newest one, but it's pretty cheap devices. Still, they can produce a lot of data. And mostly it's heavily compressed on those devices, but if you want to analyze every frame, you still have to unpack these sizes. So, handling masses of data. So, what is happening actually in your processing workflow? You start out with having the images you acquired, then you do some reduction, and you don't want to throw away your raw data, so you keep it. And then you do one step, the next step, and the next step, and every accumulates in the beginning. Actually, a lot of, so the same amount of data. Sometimes it even blows up even more between the steps because you go from 8-bit integer images to 32-bit floating point. And then all of a sudden you have factor 4 in growth in the data. And then when you come again to the segmentation, you start reducing, because when you do the segmentation, you have only, for example, black and white, and you do the labeling, you can mostly survive with 16-bit, and that's also less than you had in the beginning mostly. And finally, you come to the tables, and then it's small data to work with, and then you can work with it on any laptop. So, well, I take a lot of nice examples from SKCD. That is, in principle, the truth of it, that as soon as you start analyze, you produce a lot of data. Sometimes you can remove some of it as intermediate steps, but don't remove too much, because you may have to go back 1 or 2 steps to redo your analysis, and then you don't want to do all that stuff that you did before. So during the active analysis, you actually explode the amount of data you produce. So I have talked about terabytes, so how much is it? So let's say we have images of this type, this is just noise, 1K by 1K, and you look at them 1 each second, and then you click through them, trying to do some analysis or quick assessment, whatever. How long would it take to look through 1 terabyte if you have 1 megapixel images? I give you your help. So, doing this calculation, it's 138 hours, which is about 5 days, to look through 1 terabyte of images of this type 1 per second. So it's a lot. Humans can't do it, not reasonably. Otherwise, you have to hire a big staff of people who are clicking through images, which we don't. Then we come to the next problem. So with this in mind, we can look at images like this. That would be a more realistic example. That's actually already segmented. So you have some cells here and some other structures that are not really cells. So the task is to count how many cells you have in each of these slices, and ignore those that are too big and strangely shaped. So like this one and these lines here, they are not really cells, so those we don't want. We may also want to do an assessment. Is it more on the left side or on the right side? And are the ones to the left bigger than the other ones, or is there some relation top to bottom? And that is what we want to know from this image. One image is okay, but if you do it, you have to, in science, you need to do a lot of samples to get your statistics. So all of a sudden we got 96 of these samples. And this time it's even in 3D, so it's not so easy to just watch it. And in 3D there are 2,000 slices in each stack. So it's a lot of data, and thanks to the 3-dimensional data, we only had 2-dimensional screens, you can't do it visually anymore. And then to do it really heavy, then all of a sudden we get over 1,000 samples. First of all, how to measure it. 1,000 samples takes its time, and you don't want to run in and out. So my predecessor on this class, he actually built a robot who took the samples in and out of the tomography and did the scans. And well, when you have 1,000 of these bone samples and you want to analyze them, then you also have to have a pretty stable workflow to be able to do this analysis in reasonable time. And to make it more complicated, these cells, they are not always easy to describe. So we may have some soft metrics. So for example, how aligned are these cells? And are the group to the left more or less aligned than the ones to the right, and so on. And if you can look at these images here, you can see, well, they may be kind of aligned vertically. These are pretty well aligned. You can see everybody are going the same way, and this one, well, you guess, it's not really aligned. So these soft metrics, that is actually one of the challenges to get translated into the analysis metrics that we can get from the images. And that is actually an important skill, that you need to do the translation from the field that you're working in the vocabulary you're working in, into the vocabulary of image processing and the analysis methods. So then we can even do it even more fun. We can work with dynamic information. And we have this little movie, so it's a three-dimensional movie of some foam that is bubbling. And then it's a question about the dynamics in this sample. If you want to see where do you have small bubbles, where do you have large bubbles, which ones are moving faster, the location, where is it moving faster, along the edges or in the middle, and also rearrangement, do they merge into each other, and so on. So now, well, let's see what time is it. Okay. Now we want to bring in the scientific method in the whole analysis. So first of all, what is the purpose of the whole experiment? Yeah, we want to find and validate some new knowledge. And the question is how we can do it. And in a scientific experiment, you have to convince your peers that this actually is true, what you're saying. You can't just say, I looked at the image and say that this is actually true, but you have to show the numbers. And the other thing is that you can actually build on results from other scientists. So you have to do a lot of library work, looking in the literature and seeing what is the state of the art within the field and relate to that. And from there on, you can build up. So for this analysis, qualitative analysis is good in the beginning. It gives you an idea. You can, with your experience, you can get an idea in which direction you're going. But at some point, you have to do a quantitative analysis. And with images, you get some metrics, and the question is to find the right ones, and hopefully with sufficient certainty that you can use it for your experiment. The data may be very noisy, and things are wiggling, and so on, which makes it very hard to find the right metrics and stable ones under those conditions. And the quantitative analysis is easy in some cases. For example, if you just want to measure the temperature, it's easy. Put in the thermometer, maybe you have some error, uncertainty information about it, but afterwards you get 50 Kelvin and everybody's happy, 50 Kelvin plus minus something. But if you measure in an image, there are so many factors that are involved. Actually, the whole line of processing until you start measuring can affect the value that you are doing. And small changes in the parameterization of your filtering, for example, may make your image a little bit blurrier, which makes it harder for the segmentation algorithm to work. And that gives a lot of uncertainties. And then also, the other problem is to choose the right tool that gives you this information. And first of all, there are thousands. There are journals about algorithms for doing image analysis. And there are also thousands of tools. Over the years, I believe every student who works with image processing starts writing their own code in some sense. And there are, of course, also libraries which have been more consolidated, which today people use more, but still, there are many handmade or homemade tools. Some to solve a very specific problem. Maybe that specific problem is what you want. So then you have to go follow up on that one. And the other thing is also that images require multiple steps until you have your analysis ready. And getting to the right recipe, how to do the analysis, requires some experimenting. You have to run it on different images and see, does it work, does it not work, getting more confidence in that your processing workflow actually works the right way. And doing this experiment takes time. So it's good to have a good foundation from the literature, what is the best way of doing it. Important questions is also what you want to show. The nice renderings like this one, that's a little gold piece of gold that was, I think, was to decorate some Viking clothing or something. It looks very nice. It's very nice in presentations, but it doesn't give us any quantitative information. It just shows, okay, there we have the thing. We can look inside it. We can actually from it tell how hidden structure, how they were made or how they were put together. That we can tell, but we can't quantify it. And instead, what you can do is looking at this one, where we see a bivariate histogram of X-rays and neutrons, where you can see and follow how the gray levels depend on what material we have. So here is some soil, here is soil and corrosion, here we have ion corrosion, and here we have uncorroded iron. And then apparently we got this V-shaped histogram. And from that we can follow and try starting telling that this region was more a mixture of soil and this region is more a pure uncorroded iron. So this is more towards what we want to go. So when we start our analysis, we want to have some initial questions. So the first one is actually, what am I looking for? And put that a little bit critically. And then if you start asking that one, then is that question too general or too specific to be able to actually give you the answers that you need? And very important question is how I can plan my experiment in a way that I can do my analysis easier. It can be the order in how you do things. It can be also the way you store your data on the drive. If you organize it in the wrong way, you have a hell to really get the analysis done, because you have to write so complicated analysis scripts to find the right way where you have the data. And as I mentioned before, also how to transfer the original question into the language of image processing and image analysis. And at last, also how much a priori information do I have about the sample? So it could be, for example, the size can only be within 10 and 20 microns. If you have something that is less or greater, it cannot be the sample. It must be some misclassification or some truncated classification or something. So that is actually some boundaries and some limits that you can put in when you do your analysis. And the purpose of the experiment is also what you want to learn from it. And mostly you want to compare different treatments. You want to check different process parameters and see what they do to the sample. And you can also use it for sample inspection. And then in the end, maybe you want to do a 3D visualization because it's cool to have in your presentations. But mostly the main information is that you get tables and plots, preferably with error bars or error bands that can show how your process or how your sample behaves. What we can measure is, well, for the inspection, we can see hidden objects. We can measure dimensions. And we can count objects. We can also see mixing ratios sometimes, and we can track changes if we have a time series. So these are the things that we can actually measure from the images. And then you have to translate that into your higher level question, where you want to compare different treatments based on these metrics. So again, why quantitative? And classic example that human eyes are not so good always, because if you look at these two images and try to tell me which of the inner center squares is the brighter one, the intuitive answer would be that this one is the brighter one. Truth is, actually, they have exactly the same gray level. But depending on the context, we see different things. And this is where we can come in with our statistics. These are clean images, but usually the images always have some noise, and then you have a little bit more uncertainties. And then you have to see that the comparison between the two inner squares actually are the same. When you look at gradients, it's even harder to tell anything or do anything qualitatively. What you can see with this beamer is essentially 10 gray levels. I think we can distinguish the last two. No? Yeah, well, barely. So this is what the eye can do. I think the eye can go even up to 32 gray levels under very good conditions, but that's it. Typically, an image has 16 bits, which is more than 65,000 gray levels, and the human eye doesn't stand a chance to do anything with that. So here we really need to have the computer to tell us that these subtle changes that you can see in 65,000 gray levels, that they actually mean something. And finally, you also have to select the right tool for your task. Depends on different criteria. So for example, sorry, it's very small text. How many samples you have? That's the first choice if you want to go to doing manual clicking around, or if you want to have some automated scripting. How complex the sample is, how much human interpretation or domain knowledge do we need to put into the analysis of each image, not on the higher level, but really going into each image. What is the end product, what you need to do, and of course, can you find a tool out there that already does it? Perfect. Then you don't have to rewrite it yourself. And the last question is, you have one data set, will there be more of them? Is it worth putting the extra effort into getting a more generalized framework for analyzing these images? And the choices, depending on the answer of these questions, is going for interactive tools or scripting using existing toolboxes or also existing tools in some sense. Or finally, if those two are not available, then you have to go for developing new algorithms or writing new code. But I think that is mostly the last resort, and it goes towards this one, how much you're going to do analysis on these data. And the danger you can land in is very nicely presented here in XKCD. There is this workload. You put some extra effort in writing some code, and the automation takes over. That is the ideal case. What is often happening, you start writing a code, and then, oh, it doesn't work, so we have to do some debugging. And no, it didn't work either. Rethinking. Oh, it didn't work. And then you spend your whole PhD in developing this tool, and you lose the purpose on what you actually should do. So keep this plot in mind when you start thinking about developing new algorithms. And I think I have 2 slides, and then we take a break. And also important when you do a scientific investigation, in general actually, is there are two terms. One is reproducibility, and the other one is repeatability. The reproducibility means that two independent persons can do the experiment in the way they think, do the analysis in the way they think, and they should come to the same conclusion. This is actually pretty hard. And what we at least want is that we have experiment data, we have two persons doing the analysis, using the same tool, and the result should be the same. So it's not that there are any subjective choices that can change the outcome of the analysis. And well, I essentially already said this. And then is how we can keep track of what we're doing. It's really important when you do the analysis that you, and writing code for it in particular, is that you document what you do, and actually the whole way through the analysis. And one is keeping a logbook, preferably digital. I still see a lot of people walking around with their black notebooks and even print out things and fix them into the lab book. Please don't do it like that. Keep an electronic logbook. And then, of course, writing clear and understandable code. It's very hard to, even for yourself, if you come back to your code 3 months later, do you know what you did? It's not always so easy to understand even what you did yourself. So it's important that you write clean and understandable code if you write code. And oh, that was more than 2 slides. Okay, I think we take a break here now and continue after the break.